Today, I'm going to introduce you to the structure of DNA. DNA is a polymer of linked nucleotides. From this picture, the purple blocks are deoxyribose sugar molecules. The red circles pointed out are nitrogenous bases, which are the information carriers of the genetic material. The orange bubbles are the phosphates that are linked by phosphodiester bonds. The dotted lines between nitrogenous bases are hydrogen bonds. Let's start at the deoxyribose sugar molecules. The sugar molecule consists of 5 carbons, in which carbon 1, 2, 3, 4 are in the ring. Carbon 5 is not in the ring. The difference between ribose and deoxyribose is that deoxyribose is missing an oxygen on carbon 2. Deoxyribose is in DNA, while ribose is in RNA. They both have 1' carbon attached to the base, 5' carbon attached to the phosphate, 3' carbon is bound to the next 5' phosphate. This is done through phosphodiester bond, which is between the orange bubbles. Due to this bond characteristics, DNA strands are antiparallel, which means on the left we have 5' at the top and 3' on the bottom, while on the right we have 3' at the top and 5' at the bottom. DNA is negatively charged because of the phosphate structures on it. There are 4 kinds of nitrogen bases in DNA: adenine, guanine, cytosine, and thymine. Base pairing is very specific. A always pair with T and C always pair with G. They are important to hold the stability of DNA structure because bases are hydrophobic, so they hold together and wrap themselves up in a coil to avoid water. Base stacking energy as another molecule falls within there. It is the interaction between bases as they stack on each other. A pair with T using 2 hydrogen bonds, while G pairs with C using 3 hydrogen bonds. Hydrogen bonds are relatively weak forces, so they are not the main force to make 2 strands of DNA hold together. However, hydrogen bonds are important to know what is going acrossing from particular base and hold space between backbones. DNA helices has major and minor grooves. Minor groove is one part of coil when 2 backbones close to each other. Major grooves are where 2 backbones far apart. Watson-Crick model looks at a B-DNA, which has 10 base pair per turn and right-handed. Right-handed can be determined by the backbone close to you is up to right. Z-DNA is a left-handed, it points to the left, and it is not very common. However, we can still see many left-handed DNA structures in our daily life. Here are 5 pictures from news or books. They are all left-handed DNAs. Thank you.